Hello, everyone. It is my pleasure to welcome you to the second episode of our QBI TV series that features the women who are part of the QBI Coronavirus Research Group, known as QCRG, and who have agreed to share their reflections on what it's like to dedicate almost 2 years now of their research on exploring, understanding, and hopefully combating the COVID-19 pandemic. And with new variants on the rise, the finish line seems to be yet again pushed further away, which makes it even more essential for me to highlight the hardworking scientists who continue to fight this battle. So I'm Alexa Record, a media and events coordinator at QBI, and today we have with us a key supporter and participant of QCRG, principal investigator and virology expert, Melanie Ott. So Melanie, I now hand it over to you to further introduce yourself and tell everyone what your general areas of expertise are.
Yes, thank you, Alexa. Hello, everybody. I'm really delighted to be here. My name is Melanie Ott. I'm the director of the Gladstone Institute of Virology. I'm also a senior investigator here running a lab. And I'm also professor at UCSF and a member of QBI. You see a picture of my lab here. So we are virologists. That means we work with live viruses, including SARS-CoV-2. We built a BSL-3 specifically for work with the SARS-CoV-2 in the last year. And you see my people up there on our website working in protective gown and with respirators. In order to be protected. So we are interested in multiple viruses, classically worked a lot with HIV, but also have extensive research focuses in hepatitis C or hepatitis viruses in general, Zika virus, and have pivoted part of our lab, like other laboratories, to SARS-CoV-2 last January when the pandemic hit.
Wonderful. Thank you, Melanie. And it's always a joy for me to chat with you and pick your brain about your work, your thoughts, and particularly pertaining to this pandemic. And as you may recall, I had the opportunity to interview you right at the beginning of the quarantine in early 2020 to get your initial thoughts on not just the virus, but the incredible solidarity that we saw at UCSF and around the world when scientists began joining forces to study SARS-CoV-2 as part of QCRG. And so I want to play a segment from that interview now to sort of put things into perspective for our audience before we continue to explore your present-day thoughts. So let's take a look.
We've never had this sense of urgency that brought so many people together, and also the exclusive focus, because I have the feeling that, besides, shutting down the labs and getting everything in order and doing, what is needed also at home, I think everybody has one single focus, and that's how can we do something or contribute something that is meaningful so that we can hopefully end this soon.
That was obviously wishful thinking.
Yeah, I know. Videos like this remind me of the value of documentation, especially in times of global crises, because in this case, the past helps frame the present. And as you say, in the end of the clip, you allude to the end of the pandemic, which has still not arrived after all this time. So how does that make you feel?
Well, I think it makes me feel good, not that it's not over, but I think we are today in a much different place than we were at the time when we recorded that first video. And I think with all the hard work that scientists around the world and here at QCRG and UCSF and Gladstone has been done, I think we have been able to move very much forward in terms of new therapies, new diagnostics, and new supporting the vaccination strategies. I think that we are having some setbacks in terms of here you see a picture of one of our diagnostics that we have been developing this year, a very simple app with a cell phone where you can where you can directly detect viral RNAs in samples. And this was a great collaboration between Jennifer Doudna and Dan Fletcher and myself, and a typical example of how scientists have quickly pivoted existing technology into SARS-CoV-2, and then and now benefiting from this fast advance and support that we have received through the SARS-CoV-2 pandemic back to inform other research and other viruses in the lab. So I think there was a lot of a lot of progress that has been made, I think. And if there's something that I feel the best about, it is really the solidarity and the collaborations that have been come together so naturally in QCRG, but also with others around the world, really fast and uncomplicated, in order to see what everybody can contribute in order to deal with the situation currently. And I mean, the best example is Omicron. You see the great QCRG group here, and really, really excited to continue our common work on SARS-CoV-2, and then potentially also other viruses in the future, because that, as a virologist, is really exciting to have all these brilliant minds that have been focusing their research on potentially other diseases now also being interested in virology.
Absolutely, and you touched on this, but I also wanted to bring up that in the video you mentioned this sense of urgency that inspired so many people to focus on finding a way to contribute to the solution. And I want to know what your personal motivation was to work on COVID-19, and maybe exactly how you got roped into QCRG. What was that process like?
Well, to be roped into QCRG was very natural, because Nevin and I, we have been collaborating for a long time. We both share a very deep interest in host viral interactions, and have been teaming up in the past on other viruses. We have solved the interactome, the protein-protein interactome of hepatitis C together, and have been very closely working also on diseases like the flu together, and it was in a very complementary way, because we have we can contribute some virology. We can also contribute some of our cell culture interests that we have with specific cell cultures called organoids, where we use primary 3D cultures of cells. Here you see lung cells or intestinal cells, and then with colleagues here at Gladstone, we have also worked on heart cells that we can infect and then follow infection in the cell, like you see on the bottom, all the green dots, and on the right side in the heart cells, the orange dots. These are all viral replication centers that we can visualize and then study how this virus really intricately interacts with these different cell types. And so I think for me, it was a no-brainer to team up with QCRG and help with whatever we have, and continue the long-standing deep collaborations that exist between the Ott and the Grogan labs.
It's really fantastic. And I think also your area of expertise sort of makes it a no-brainer for you to join this effort. But I want our audience to be on the same page as us, so I'm going to ask you a very basic question. Can you describe the role of a virologist, and what does studying viruses really entail?
Well, rule number one today in the pandemic is that everybody is a virologist now, which is great, because everybody understands basics of viral replication, transmission, spread, resistance development, mutation development, variants. All these are things that we deal with on a very daily basis. What maybe distinguishes us from others is that we are really working with live viruses. As I said at the beginning, we work under high biosafety containment conditions, like the BSL-3, where we really have to use protective gear in order to be able to be exposed to the virus that could potentially infect us ourselves. And you see another picture here of the crew in protective gear, really at this point culturing the first aliquot that we had of the virus of SARS-CoV-2, and really eagerly looking under the microscope if that virus is inducing cell death in the culture, which is indicative that there's really something growing. And it's very much similar to what's happening now with Omicron here in San Francisco, because you get a sample from a patient, you put it in culture, and you have to make sure that that virus is somehow infecting the cells in the culture so that we can generate more virus and can use it for studies both in cells but also in mice. And I think the environment that you're seeing here is highly regulated. It's extremely safe. So I would say the work in the lab is the smallest part. I think the regulatory and the training part to get into the lab is enormous in order for us to do exactly precisely experiments that we map out in advance in order to do them in a safe and controlled way.
It's always inspiring to see scientists in action. That's a fantastic picture that you dug up. And when I last interviewed you at the start of the pandemic, you had a very focused goal. And you said it was to disable SARS-CoV-2 in the cell and identify its natural restrictive factors. And as science progressed over the last almost 2 years now, I wonder, how has your research focus expanded and maybe broadened?
Yeah, I think when we deal with a new virus like SARS-CoV-2, we have to see what we can offer, what we can apply, and how we can make a meaningful contribution. And we also have to see what is the immediate questions. I think for us, the immediate question about SARS-CoV-2 is how can we interrupt it? How can we get ahead of it? And I think the approach that QCRG has taken was a very smart one, because we know that viruses are critically dependent on the host cell. So viruses, contrary to other like bacteria, really need the host essentially in order to throw the virus lifelines in the cell in order to replicate. So without us, the virus cannot replicate. It has to jump from cell to cell, from person to person in order to survive. And so to disrupting this dependency of the virus on the host cell, or to go after these critical lifelines, is really an immediate way to go after viral infections. And it turned out the work from QCRG and from the Krogan lab and our lab has really shown that these lifelines are very similar sometimes, but between very different viruses. Every virus is particular, and it goes into different cell types. It has a unique way to replicate, but they all need energy. They all need membranes. They all need certain components of the cells in order to multiply. And so to go after these critical conserved pathways that are absolutely critical in host cells, I think is one way to deal with viruses fast. And I think QCRG has done this beautifully by identifying, the host interactome and particularly identifying certain host factors where already existing drugs are approved, so that we could use these drugs immediately to see whether this would diminish replication of the virus and potentially disease and severity of the infection. And so I think this was our immediate goal. I think we have since also broadened our interest. We're still critically interested in these host viral interactions, and we have really only scratched the surface. We're learning every day about new things that the virus is doing, but there is also, as I said, other things that we are interested in. We had, for example, a diagnostic that we have been developing for HIV, what we saw at the beginning with a mobile phone and CRISPR technology, which is really a new way of detecting viral RNA directly without reverse transcription and amplification like we do now with the PCR assay or other molecular testing strategies. And because it was so difficult at the beginning to get to tests, I think we pivoted this technology that we developed for HIV into SARS-CoV-2, and that has now accelerated the development of the technology to the point where it's becoming applicable for the community for SARS-CoV-2, but also is back informing us, our research in the lab on HIV diagnostics and other RNA detection systems. So we're really excited about various ways how we have broadened out. We're very interested also in the way the spike protein can evolve, and to really find out what makes a variant highly transmissible and what makes a variant immunoevasive. We're very interested in studying the virus in the different in the different organs that it can infect. The lung is definitely its primary target, but we know that there's also a huge gut involvement, and we have shown, and many others have shown that the virus has actually tropism for quite a few organs, and we see long-term effects in many of these organs. So there's a lot that we have not fully understood. We have also broadened our BSL-3 to include animal research. So we're doing now research in mice. And that can help us to learn a lot more about how people are getting sick and how viruses are making people sick, and how viruses can also be transmitted from one individual to another. So there's a lot more research ongoing here, but as we see, the pandemic keeps a lot of surprises, for us in store. So we better be prepared to tackle them in the future.
That's right, Melanie. And if there's one thing that I've learned here at QBI about scientific research, is that it's all connected. It's all, intertwined in some way or another. And so thank you for highlighting how, how COVID is helping us make sense of other diseases, and how at the end of the day, everything infers all sorts of research areas. To sort of pivot, I wanted to ask you about some of the challenges that came up during the pandemic, because living and working during a pandemic is taxing in many, many ways. So what are some of the challenges you and your lab face, and maybe how did you navigate those?
Yeah, how about working under a lockdown? I think that's probably the challenge that everybody faced. I think it was an interesting time at the beginning. I think our biggest challenge was the BSL-3. We did not have our own airborne pathogen BSL-3. We work a lot with bloodborne pathogens, and we were very well equipped for that. So colleagues at UCSF helped us out to first get into the BSL-3 there until we had ours ready. But again, an amazing feat in getting people together, engineers, regulatory people, everybody was pitching in, driving at night from Irvine up in order to be here to certify our BSL-3. So an enormous effort and collegiality that came into it, but really important, because as you see, this is really the life space for a virologist. So if we don't have the opportunity to really work with it, it's hard. So I would say the BSL-3 and lack of BSL-3 and shortage is something that was imminent for many people around the world, and I think there's a big push to expand this for the future, to be better prepared for future pandemics. But other than that, I mean, I think the lockdown conditions, but also moving from one building to the other, we had to drive to Parnassus with all our equipment, with all our materials and supplies and cells in order to work in the BSL-3. We had wonderful hosts with Anita Seal's lab who were waiting for us at the door to let us in, to be with us the whole time, and then close the door behind us again because everything was in lockdown, in order to receive materials and supplies, and even very very important issues like viral stocks or anything, very secure packages. We had to make sure that we are on the minute meeting with the person at the right spot, because there was no receptionist or anybody who could receive it, no receiving person on site. So I think all these like little logistic things that can take a lot of time, but are very important because it was quite challenging at the beginning to work, and now we are luckily back to normal, and that I think helps enormously. But one forgets. I think the biggest challenge that we still face is the shortage of supplies. PPE was extremely, which is personal protective equipment. It was very, very in short supply at the beginning, especially for these respirators. Also, we had to make sure that we really got everything that is on the market, and everybody else was trying the same. So we're still having shortages. Everybody knows this, tips, pipette tips, or other things, because of the general shortage that is currently ongoing. But at the beginning, very, very, very difficult to get PPE. We had to make sure that we reuse everything safely, gloves, masks, all of these things we forget, but was something that was in short supply.
Definitely. I guess we get smarter with time. We get more used to it with time, but the challenges certainly teach us many lessons along the way. I think it's also important to focus on the positive aspects that come with spending countless hours, months, years working on understanding SARS-CoV-2. And you've mentioned several already, but I wonder, what were some of the key contributors that helped you answer your research questions? You mentioned the BSL-3 lab. That was a very big breakthrough, or being able to include mice in your studies. Is there anything else you'd like to add on the breakthrough front?
I think we have made enormous progress in many ways. I think we, as virologists, you know how to handle viruses, but you have to get used to specific viruses. This was a new virus, so we have learned a lot about how we can culture the virus, how we can safely handle the virus, and how we can infect, all these different cell types that I've showed you and the mice. Mm-hmm. I think one big breakthrough that we made recently is, I would say, as a molecular virologist, where you want to go very much into detail, you want to also manipulate the virus. So we're not just, working with viruses that we isolate from patients and that culture and that keeps in culture. And when they keep in culture, they actually keep changing, because you have to be very careful that they're not adapting to the culture conditions, but maintain their original phenotype. And also accumulate more mutations. So I think what as a virologist, what we do is we reconstruct viruses, build basically a copy of the original virus that we can then put in culture very freshly, and then we can also introduce, for example, mutations from the variants into that, and can study how these individual mutations are really affecting all of the different parameters that I have told you. So I think these are again very highly regulated processes that cannot be done just everywhere with whatever we want. I think they're very highly regulated because they need to be done safely. But I think this is probably our best bet in order to get ahead of this virus, because we will really understand now the many mutations that Omicron has in much, much better detail. And so this for us was a recent breakthrough, that we can do this now very rapidly and very safely.
Wonderful. Wonderful. And while we're on this topic, I want to bring up an audience question. Here it says, you spoke of managing surprises. Do you think scientists can predict new variants and their behaviors? Can scientists get better at that? It's a great question.
Absolutely. I think a lot of people are doing this. I think we are doing this. I think we have now, and this is a very big collaboration between computational experts and wet lab experts, because I think you have to somehow really use AI in order to learn from what is existing to potentially predict the future. Mm-hmm. We have a problem here, and we can do this safely if we look, for example, just at the spike protein, and we can make surrogate viruses just carry the spike protein but have otherwise nothing to do with SARS-CoV-2, and then we can safely introduce many mutations and test them. When it comes to the full-length virus and we generating new viruses, I think we have to be much more careful in order to not manipulate the virus in a way that it becomes better than the original virus. So I think this is definitively something that is ongoing in many laboratories, including ours, but it has to be always done with the safety and regulation in mind first.
Mm-hmm. Definitely. And while we're on the topic of variants here, I just want to take a few more minutes to highlight how COVID-19 is going to keep being part of your daily routine almost 2 years later. What is being done so far to work on the Omicron variant, at least at UNICEF, or how has that been for you?
Well, Omicron comes after we had Epsilon, after we had Delta. So I think for us it's relatively familiar because we have been here in the Bay Area. The Epsilon variant was the so-called California variant, which was circulating primarily here in California. Now Omicron was first detected in the US here in San Francisco, so that gives us the opportunity to actually culture the virus and study it. So there's a lot that's currently being done. I think the most pressing question is for everybody is that what is the response to the immune serum of this virus? And there is and it's probably currently being studied in South Africa with the colleagues there, in New York and here and everywhere around the world. I think there will be very similar experiments being performed. I think here in the Bay Area we have a wonderful consortium of the individual BSL-3 that are available at UCSF, but also at Stanford and at Berkeley and at UC Davis. We meet regularly, we exchange experiences, we share reagents in order to, synergize and not duplicate our work. So there's everything that has been done before is now going to be thrown on the Omicron virus in terms of, or variant in terms of questions. As I said, our toolbox has enormously expanded over the last 18 months, which is great because we can react much faster and much more directly to these evolving variants.
Definitely. And from what you're saying, I'm gathering, again, the importance of collaboration in science. It's been a big part of QCRG. I think it will continue to be as you study variants to come. So this sort of segues into our next audience question about collaborations here. Do you think that COVID-19 has actually changed how scientists work together? Or do you think that once the pandemic is over, so will the collaborations?
I hope not. I think this is really one of the most positive outcomes that we have of this pandemic is, as I said, feel the urgency, feel the need to work together just because we need something now, not in 3 years or in 5 years or in 10 years. I think that it has changed how we work together. I think we not only among virologists, but also among every other scientist, because I think when the pandemic hit, everybody tried to contribute or to see what they could contribute meaningfully. And I think that made it so immediate and so urgent and so successful. And QCRG is the best example because we have, all of the fantastic basic scientists that are at UCSF in there sharing their expertise, applying it to this virus. So I hope this is not going to end. I hope everybody will maintain to be a little bit of a virologist in their heart. Because we all know that it's important. It can happen again. And first we have to dig ourselves out of this one.
Thank you, Melanie. I really hope so too, because it's been proven to be a recipe that works in many, many ways. So I hope that collaboration continues to be part of our focus here at UCSF and beyond. And with that, it looks like we are nearing our 30-minute mark already. So I'd like to wrap it up by thanking you, Melanie, again for being part of this series and opening up to all of us about your journey at QCRG thus far. And again, I commend you and all of the hardworking people who continuously strive to make a difference in healthcare. And I'd also like to thank everyone who tuned in to this episode. I welcome you to stay tuned and also watch the previous ones featuring Shari Mukherjee. I look forward to welcoming you for our next one early next year. So subscribe to QBI TV for more, and thank you again for being with us today.
Thank you, Alexa. Thank you, everybody, for your attention. Bye.
Bye-bye.